Hello, everyone. Thank you for joining us for our Celpress webinar in these extremely challenging times with COVID-19. I hope you are all doing well. This webinar is on chemical probes as essential tools for biological discovery. I'm Mishti Day, editor at Cell Chemical Biology, as well as the journal Structure. I will be your moderator for today's webinar. As you know, chemical probes are powerful tools to interrogate complex biological systems. The probes have facilitated key discoveries that range from translational research to basic understanding of how cellular signaling processes work to developing targeted therapies. High-quality small molecule probes are complementary to commonly used genetic strategies and are central to basic and translational research. This webinar is aimed to provide researchers involved in developing and using chemical probes with practical suggestions to help select the most appropriate research tools along the path to amazing discoveries. Our experts today will also share how innovations using chemical probes are helping to enhance our understanding of complex disease pathways and mechanisms and accelerate translational efforts. In this Celpress webinar, we will discuss the latest breakthroughs and technical advances in chemical probes for biological discovery. We are excited to have with us today three outstanding experts in the field of chemical biology. Daniel Nomura from University of California, Berkeley, Anna Greca from Brigham and Women's Hospital and Harvard Medical School, and Paul Workman from the Institute of Cancer Research in London. Before we get started with the talk, we have a few practical points that I would like to share. Each of our speakers will talk for approximately 15 minutes. Following the talks, we'll have a Q&A session that covers all three presentations. If you have a question for any of the speakers, just click on the question tab at the top of the display and type it in. You can also enter a question at any time. No need to wait until the end. We will try our best to pass on as many of your questions to the speakers as we can during the Q&A session. Before we begin, we would like to thank our speakers for making time to join with us today, and also our sponsor, Cayman Chemical, for supporting this webinar. Celpress also offers a free resource for R&D professionals called ResearchArc. Sign up for monthly access to peer-reviewed research from Celpress that mirrors the trajectory of drug development at www.cell.com/research-arc. And now, I will pass the ball to our first speaker, Daniel Nomura. Dan, thank you for joining us today.
Thank you, Mishtu. Thanks everybody for logging in. So I'm going to be talking about some of our lab's recent work on tackling the undruggable proteome using chemoproteomic platforms. These are my disclosures. Our lab is very much interested in tackling the undruggable proteome. I think this is probably one of the biggest rate-limiting steps in drug discovery right now, that over 90% of the proteome is currently considered undruggable because most proteins don't really possess known binding pockets that small molecules can bind to affect their function. So unlike druggable proteins like enzymes and receptors that have nicely formed binding pockets that a small molecule can bind to, undruggable proteins include things like protein complexes or intrinsically disordered proteins, where it's just not really clear where you can find a pocket within some proteins like this. Classic examples of undruggable proteins include things like KRAS, c-Myc, p53, and p21, which we've known for decades are major drivers of or tumor suppressors of cancer. Yet, except for the case of KRAS G12C mutants, with the help of Kayvon Chokat's work at UCSF and now many other pharmaceutical companies that have developed KRAS G12C inhibitors, other mutant forms of KRAS or direct inhibitors against c-Myc or p53 direct binding activators or p21 activators still remain quite intractable for drug discovery efforts. And these four proteins are really just the tip of the iceberg of tens of thousands of other protein targets that are still thought to be quite intractable for disease therapy. And so our premise is that if we can identify binding pockets or druggable hotspots across all proteins in the proteome, then we can potentially go after developing small molecule therapies against any protein target of interest. And so to do this, our lab utilizes chemoproteomics, and we couple that with covalent ligand screening to both map and pharmacologically target druggable hotspots in complex living systems. We also take lessons learned from natural products that are often able to access really unique therapeutic modalities, and so we try to learn from how natural products do that and exploit that with simpler chemistry. And we're also very much involved in the space of chemically induced proximity, which include things like proteolysis targeting chimeras or PROTACs for degrading specific protein targets of interest, but also, for example, developing new molecular glue strategies to be able to bring proteins that usually wouldn't interact into close proximity towards conferring neomorphic functions, all towards discovering new therapeutic targets and drugs. So the underlying technology that our lab uses is an approach called activity-based protein profiling, or ABPP, which was originally pioneered by my former postdoc mentor, Ben Cravat, at Scripps. And this technology uses reactivity-based chemical probes to profile proteome-wide reactive functional ligandable sites directly in complex biological systems. And the way this technology works is that you have a series of electrophilic reactivity-based probes that can covalently modify nucleophilic amino acid hotspots within proteins, such as serines, cysteines, and lysines that not only take place in enzyme catalysis, but also post-translational modification sites, protein-protein interaction sites, allosteric binding pockets, or other types of binding modalities. And you can read that binding event out because these probes bear an alkyne handle. So you can use copper-catalyzed click chemistry to append on your analytical handle of interest, whether it be a fluorophore like rhodamine or an enrichment handle like biotin. And so showing you here various examples of reactivity-based probes that the chemical biology field has made through the years for example, serine, cysteine, lysine, and methionine reactivity, and they all bear these red reactive warheads and blue alkyne handles. So in a typical experiment, you can take any kind of complex proteome and you can throw in your reactive probe. The probe will react broadly across thousands of ligandable sites, and you can read that out by clicking on a cleavable biotinazide tag onto your probe-labeled proteins, enriching your proteins, digesting your proteins, and releasing your tag to just give you an enriched pool of tryptic peptides that are bound to your probe and to the little mass tag. And you can shoot that on the mass spec to identify all the sites of probe labeling. And all of these sites of probe labeling now potentially constitute ligandable sites that can now be pharmacologically interrogated.
That's great. Got it.
And collectively, hello?
Okay.
Collectively, we've identified upwards of over 100,000 ligandable sites over 16,000 human protein targets. So when people say that most proteins are quite intractable because we don't know where the pockets are, we've identified potential pockets across most human protein targets of interest. And so to be able to interrogate those as small molecules, we and many others in the chemical biology field have built out large chemical libraries that possess these reactive warheads that we can use to interrogate against those sites. And so, for example, if you find a cysteine reactive hotspot with a broad promiscuous iodoacetylide cysteine reactive probe, we built out thousands of these small molecules that all bear these acrylamide or chloroceramide cysteine reactive warheads, and by diversifying the R groups, you can confer specificity for certain cysteines over others. And so you can screen these libraries in a pure protein target basis, where you can, for example, screen a covalent ligand library against your pure protein, and if it binds compared to your control, when you chase with the reactive probe, the probe can no longer bind, and so you can read that out as loss of fluorescence. But most importantly, you can do this directly in complex living systems, where you can envision treating your covalent ligand directly in vivo in a mouse, or in situ in cells, or in vitro in proteomes, and your ligand will bind to whatever targets it's going to bind to. So when you chase with your reactive probe, the probe can no longer bind to those liganded sites anymore. So it's a simple competition experiment, but across thousands of protein targets. And you can quantitatively read that out by clicking on isotopically light and heavy mass tags onto your probe-labeled proteins, and then taking it through this whole process. And now identifying the specific sites for your small molecule ligand displaced probe labeling, enabling you to identify exactly which amino acid and which protein target your covalent ligand bound to directly in a complex biological system, and allows you to assess how selectively it bound to that target on a proteome-wide scale. And so what have we been doing with this? We've been screening these covalent ligand libraries and various target-based biochemical and phenotypic screens. And once we identify hits, we can deconvolute mechanism of action using chemoproteomic approaches. And so I'll just show you two quick vignettes around this. This one first is going after discovering new activators of autophagy. Autophagy is a really important cellular quality control program, which is involved in eliminating toxic protein aggregates or misfolded organelles. Aggregated protein aggregates or damaged organelles, and it does so by engulfing that cargo into autophagosomes and then fusing with the lysosome, and then subsequently being degraded by the lysosome to recycle nutrients. And autophagy is dysregulated by a variety of diseases, and it's thought that if you could activate autophagy, you could potentially treat a variety of disorders, including cancers or neurodegenerative disease or atherosclerosis or even things like aging. And so to address this, we screened our cysteine and lysine reactive libraries in a phenotypic screen to look for things that could activate autophagy, using this dual color reporter assay, looking for which reads out basically the degradation of this autophagy adapter protein called LC3. And we screened in both mouse embryonic fibroblast cells and also took those hits and screened in human cells. And the best hit out of that was the cysteine reactive acrylamide, EN6. And so we wanted to figure out how this worked. And so we treated cells with EN6, and then we chased with our cysteine reactive probe and used chemoproteomics to figure out which target this was binding to. And it turns out that the primary target that this is binding to across over 1,000 cysteines that we're profiling here in this case was the cysteine 277 on this protein called ATP621A. And it turns out that this protein is part of the VATPase catalytic subunit that is at the lysosome that helps acidify the lysosome. And a close collaborator and good friend and colleague of mine at UC Berkeley, Roberto Zancu, had discovered that this ATPase actually acts as an amino acid sensor, and that under plentiful nutrients, it recruits this regulator RAG complex to recruit mTORC1 to the lysosome and to subsequently activate it. And when mTORC1 is active, that subsequently inhibits autophagy to stimulate growth signaling pathways. So we thought that our molecule EN6, through binding the VATPase, disrupts this regulator RAG complex to disrupt mTORC1 localization, thereby inhibiting its activity, subsequently activating autophagy. And so this was a collaboration done with Roberto Zancu's group at UC Berkeley, where we were able to show that when you stimulate cells with amino acids, all the mTORC1 localizes to the lysosome, but when you treat those cells with EN6, mTORC1 no longer localizes to the lysosome, and that leads to the complete and selective inactivation of mTORC1 signaling, as you can see by loss of phospho-S6K and ULK1. But unlike other mTORC1 inhibitors, such as Torin-1, which have off targets, such as inhibiting AKT signaling pathways, our molecule actually does not inhibit AKT pathways. And also, unlike rapamycin, which is only an incomplete inhibitor of mTORC1 signaling, it seems like our molecule is actually completely inhibiting mTORC1 pathways. So if you mutate that cysteine to an alanine or a serine, then subsequently we get complete resistance to EN6 effects, further speaking to the selectivity of this molecule. And that subsequently leads to downstream improvement in cellular quality control. So if you take this kind of artificial cell model where we can induce with IPTG these TDP-43 aggregates, which are found in ALS and FTD, you can see that EN6 is robustly able to clear those toxic protein aggregates. And if you treat those cells with a VATPase, an autophagy inhibitor, rapamycin, you can actually prevent that clearance. And quite interestingly, even though this molecule is not that potent, although it's quite selective, it's already in vivo active. And in fact, it completely shuts down TORC1 signaling in vivo to the same extent as rapamycin. But unlike rapamycin, which is, again, only a partial inhibitor of mTORC1 signaling, EN6 is able to completely shut down 4-ABP1, ULK1 pathways in vivo in mice, and also activate autophagy in vivo as well, as shown by the P62 degradation at the bottom of that plot. And so this is a quick example of how we can take a molecule like EN6, or do phenotypic screening with our covalent ligand libraries, discover a molecule like EN6, which is able to activate autophagy through this unique mechanism of targeting this novel ligandable site within the VATPase, which then disrupts the mTORC1 localization to the lysosome and subsequently inhibits its function to subsequently activate autophagy. We also have a longstanding interest in natural products, which oftentimes are able to access really unique therapeutic modalities. And that we would usually consider undruggable. And so we try to learn from natural products and then try to exploit that same type of modality with simpler chemistry. And we tried this out with this natural product called nimbolide, which is derived from the neem tree, which grows in India. And nimbolide is one of the main components of the neem leaves, and it's been shown to have anti-cancer, antibacterial, and pesticidal activity. But the mechanisms have really not been understood. Nimbolide has this cyclic enone in red here, which can potentially covalently react with cysteines. And in our own hands, and it's been shown by many others, that this is able to compromise breast cancer cell proliferation. And with chemoproteomics, we figured out that the primary target of this was a selective covalent modification of this N-terminal cysteine 8 within the E3 ubiquitin ligase called RNF114. It turns out, interestingly, that this cysteine 8 belongs to a predicted intrinsically disordered region within RNF114, which is usually thought to be impossible to develop a ligand against. But nonetheless, through a lot of biochemical studies, we've been able to show that this, in fact, binds to this intrinsically disordered region within this E3 ligase. And this is, in fact, the reason why the cancer cells are dying. If you knock down RNF114, you get resistance to nimbolide-based anti-proliferative effects. And so what does RNF114 do? Again, it's an E3 ubiquitin ligase that recognizes tumor suppressors like P21 to ubiquitinate and degrade those targets by the proteasome. And so when we reconstituted the activity of this enzyme, we could show that both the autoubiquitination and the ubiquitination of the substrate P21 by RNF114 was completely inhibited with nimbolide. So we initially thought we had an inhibitor of this E3 ligase. And when we did quantitative proteomics on breast cancer cells that were treated with the nimbolide, we got elevations in the tumor suppressors P21 and P57, which are known substrates of RNF114, as well as these two novel substrates of RNF114, PEG10 and CTGF that we discovered here. But we nonetheless thought that elevation of these tumor suppressors was leading to its anti-cancer effects, and consistent with that, if you knock down both P21 and P57, you attenuate the anti-proliferative effects conferred by nimbolide. And subsequently, using biochemical studies, we found that we're not actually inhibiting the function of RNF114, but rather its ability to recognize its substrates like P21. And that becomes quite critical on the next slide. But we're not actually inhibiting RNF114 activity, but disrupting a protein-protein interaction between RNF114 and P21. And that's really important of a distinction, because we thought that maybe we could actually use nimbolide as a novel E3 ligase recruiter in targeted protein degradation applications, which is a huge area in drug discovery right now that was pioneered by folks like Craig Cruz and many others, where it uses these heterobifunctional degraders, where basically you have a small molecule that binds to a protein target of interest that's linked to an E3 ligase recruiter that can subsequently recruit an E3 ligase to ubiquitinate and degrade your target. And so in collaboration with Tom Maimoni's group at UC Berkeley, we were able to make this degrader molecule where we linked nimbolide onto the BRD4 inhibitor JQ1, and we can subsequently show that we could degrade BRD4 within cells, and that was prevented by a proteasome inhibitor. And with quantitative proteomics, we were able to show that we could selectively degrade BRD4 in proteomes. You also still get elevations in the endogenous substrates of RNF114, like tumor suppressors, including P21 and P57. But in the context of cancer, we see this as a positive, because you're eliminating a oncogenic protein while stabilizing tumor suppressors. And just to show you this is on target activity, if you knock out RNF114 with CRISPR-Cas9, we can in fact show that we can completely attenuate the degradation conferred by this XH2 degrader that you observe in wild-type cells. So now that we know, right, that this very complex natural product, nimbolide, is acting through targeting RNF114, we can now try to develop a much more synthetically tractable small molecule that does the same thing. And so we now took purified RNF114, we screened our cysteine reactive library against it, and developed EN62, which is a cysteine reactive acrylamide, much easier to synthesize than nimbolide, which shows exactly the same activity. It inhibits autoubiquitination activity of RNF114. It selectively targets that cysteine on RNF114 on a proteomoid scale. It's able to compromise breast cancer cell survival. And we can show that we can give it to mice once per day and significantly attenuate breast tumor growth in vivo as well. And so this is just a quick example of how we can use chemoproteomics to deconvolute the mechanism of action of a natural product like nimbolide and exploit that same modality with simpler chemistry, for example, with EN62. And we can also use nimbolide as a novel E3 ligase recruiter for targeted protein degradation applications. And so we've been able to add upon, one of the kind of rate limiting steps of this technology has been the lack of E3 ligase recruiters. And we've been able to add upon that by adding on Nimbolide as an RNF114 recruiter. But certainly that was kind of a fortuitous way of discovering a new E3 ligase recruiter. But in fact, we've identified ligandable sites across 450 of the 600 putative E3 ligases. And so we can now systematically go after developing new E3 ligase recruiters for targeted protein degradation applications. And in fact, we've done that already, and we've developed several new E3 ligase recruiters that you can read about some of them in published literature and on bio or on Chem archives. Looking forward, we're also looking at different types of heterobifunctional modalities beyond just recruiting E3 ligases. We're now looking at, for example, recruiting deubiquitinases and other types of enzymes to do surgical chemistry on protein targets. And also we're developing new approaches using chemoproteomics to discover new molecular glue scaffolds to bring two proteins into close proximity that usually wouldn't interact to be able to confer geomorphic functions to proteins. So hopefully I've convinced you of the utility of chemoproteomics in tackling non-druggable proteome, and I'd like to thank my lab at UC Berkeley, as well as our collaboration with Novartis, which has been a really critical key collaboration that we've had for several years now, where basically all of these stories that I talked about today were actually supported by our Novartis Berkeley Center at UC Berkeley to use chemoproteomic approaches to tackle the undruggable proteome, and also the Mark Foundation for also helping to fund this research, and also the Innovative Genomics Institute at UC Berkeley. And I'll now hand it over to Anna Greca.
Thank you, Dan. This was a great talk. Now we are going to move on to Anna Greca. Anna, thanks so much for being with us today.
Thank you for having me, Mishtu. It's great to be here with Dan and Paul. I'd like to switch gears a little bit and consider a different approach and maybe a different set of diseases. So these are my disclosures. I would like to provide a framework for how my lab likes to think about basically everything that we do. The focus of our work is really centered on going from genes to mechanisms to medicines. So we believe that genetics and genomics has reached a pivotal time when we have an abundance of information that we can now harness to do deep mechanistic work that will ultimately reveal what I think is a good as any definition of precision medicine, meaning matching the right patient with the right treatment at the right time. And so argument could be made that success in this area might be easier had in the case of Mendelian monogenic disorders. This is the case in which a singular gene contributes to disease, and that provides an opportunity for very detailed mechanistic work in relevant cell types that can ultimately lead to precision medicine. So this is an area where my lab has had significant interest for a long time. We're essentially cell biologists who like to start with genetics and then use every tool available to us, whether genetic or chemical, in order to illuminate mechanisms that will ultimately lead to therapies. And one area in which we have devoted considerable attention in recent years has been a rare autosomal dominant kidney disease called mucin-1 or MUC1 kidney disease. The causal mutation was identified some years ago by Mark Daly and Eric Lander's groups at the Broad Institute in the MUC1 gene, the mucin-1 gene. And so the task at hand for us as cell biologists was really to understand the underlying molecular mechanisms in the kidney to ultimately hope to develop a path toward a therapy. Just by way of background, MUC1 kidney disease is again an autosomal dominant disease that affects the tubular compartment of the kidney. It is rare and it presents in a very bland way, such that most kidney specialists even have trouble recognizing this disease. There are no available treatments. And the mutation, while the general vicinity of the mutation or the haplotype was known for about 20 years, it actually took very significant effort at the Broad Institute to eventually identify it as a cytosine insertion that causes a frame shift in the variable tandem repeat region or VNTR region of the mucin-1 gene causing essentially a frame shift and an early stop codon, as shown here on the bottom, that results in a neoprotein. This is the NTR region of this gene is essentially what one could call a very dark corner of the human genome, one that required special efforts in order to identify this single letter insertion, the plus C insertion that causes this trouble. As you can appreciate from the graphical representation, this neoprotein lacks the transmembrane domain. It no longer functions as a regular mucin, a protein that has a transmembrane domain and is able to insert into the plasma membrane and produce mucus, which is an essential barrier function for all epithelial cells. And it essentially becomes what was initially a mysterious neoprotein that no one knew what to make of. So this is the beginning of our story in some sense. A very talented postdoc in my lab, who will soon be an assistant professor, started with the question of how could we start to model this disease so we can begin to understand what's going on. And Moran developed a MUC1-FS knock-in mouse model. So we essentially removed the normal, one of the two normal alleles from mucin1 from the mouse and replaced it with the human mutant allele. We also generated a control mouse that had the wild-type version of the human allele, which did not develop any disease. But we were pleased to see that the knock-in mouse for the mutant version developed a disease that seemed very similar to patients, as shown on the bottom panel, suggesting that we were on the right track. What is shown here is essentially a kidney that develops these holes, what can be appreciated on the histology, which are called dilated tubules. These are essentially kidney structures that can no longer function as regular tubules, and that ultimately leads to kidney failure. Armed with this, we actually raised a specific antibody that recognizes the mutant version of the protein, as shown here in green. The wild type will be shown in red. And as you can appreciate, the areas where the kidney shows the most abnormalities, these black holes as shown here, are also areas of very high expression of the mutant version of the protein, again suggesting that we were on the right track. So armed with this, we wanted to understand what are the sequelae of this mutant protein that accumulates in these tubular epithelial cells in the kidney of these mice. And what we found, of course, is that the accumulation of this protein causes increased apoptosis, as shown here in the red dots and quantified here. And this is a time-dependent phenomenon, meaning early on, we don't observe apoptosis, but as the mice age, we increasingly observe cell death, suggesting that MUC1 kidney disease, or MKD, is essentially a toxic proteinopathy. In work that I do not have time to show, we did detailed analysis to show that the accumulation of this toxic neoprotein essentially triggers the apoptotic arm of the unfolded protein response that ultimately leads to apoptosis through an ER stress pathway. Armed with this understanding that we were dealing with a toxic protein, we of course wanted to develop a therapeutic hypothesis that would involve the removal of the mutant protein from these kidneys. But in order to gain more confidence in our system, we wanted to make sure that what we observed in the mouse models or in the cell-based models was actually true in the human as well. So through work of Mahesh Amani in the lab, we developed kidney organoids, which were derived from pluripotent stem cells kindly donated by patients with this disease. Their unaffected siblings were used as controls. And as you can appreciate here, what I'm showing you is basically the tubule, focusing on a tubule of such a mini kidney in a dish, if you will, from a patient with the disease. And what you can appreciate is that the mutant protein, again in green, is accumulating haphazardly in the intercellular compartment, while the wild-type version of the protein, remember this is an autosomal dominant disease, so patients have one normal allele, is appropriately apically localized in this tubular epithelium cell or collection of cells in this tubule. So we actually had evidence that this toxic protein accumulation was not only relevant to mice, but was also relevant to a human-like system. We also were able to develop a tubular epithelial cell line, an immortalized cell line, kindly donated from a patient with the disease. And again, you can appreciate that the wild-type protein is appropriately apically accumulated, while the mutant protein accumulates intercellularly. This tool was actually instrumental in what we wanted to do next, which is actually to screen a library of compounds, with the therapeutic hypothesis being that we wanted to find a compound that removes the toxic protein from the intercellular compartment, the accumulated protein. And we did exactly this. Based on time, I'm going to be fast forwarding through a lot of this work that we did. We basically screened a collection of all known FDA-approved compounds and several preclinical compounds, what is known as the broad repurposing set, and identified a preclinical compound, a compound that has never made it into a human, called DRD4780. Its structure is shown here. And as you can appreciate, and this is elegant work done by Moran and Masha in the lab, the compound caught our attention in this high content screen that we did based on confocal microscopy, looking for the removal of the mutant protein, because it actually did exactly that in a very nice dose-responsive manner with a nice EC50 around 150 nanomolar. And it did so without any appreciable cellular toxicity and without affecting the wild-type version of the protein. And so it fulfilled a lot of criteria that piqued our interest in this compound as an interesting tool for further study. We, of course, wanted to make sure that this tool works in vivo, and so here's the experiment in the mice. Within 1 week of oral gavage of these mice, here's a control mouse expressing only the wild-type version of the protein. Here's a mutant mouse expressing large amounts of the mutant protein, and here's a litter mate of that mouse expressing the mutant protein, but having received drug for a week. And as you can see, there was a very effective removal of the mutant protein from these mouse kidneys. We, of course, wanted to see whether this was true in patient organoids. And so these are 3 patients who donated their organoids. And one of the siblings shown as a control. We had paired siblings for each of these patients. And as you can appreciate, again, the intercellularly accumulated protein was removed. This is after 3 days of treatment with the BRD compound in vitro in these human mini kidneys in a dish. Pleased with this efficacy for our compound, we of course wanted to answer the fundamental question, which is how does this compound work? In order to do this, we had to do some detailed cell biological work to truly understand what is the intracellular compartment in which the mutant protein accumulates in the first place. We went through a detailed imaging experiment to actually find that ultimately the majority of the mutant protein was accumulated in the COP1 compartment. This is a vesicular compartment that connects the cis Golgi with the ER, and its job is to basically return either misfolded proteins or resident ER proteins back to the ER. We found that the majority of the mutant protein accumulates in this TMED9 rich vesicular compartment between the Golgi and the ER, although it has the capacity to actually traverse through the secretory pathway and indeed reach all the way into the lysosome in small amounts, even under baseline conditions, but clearly not sufficient to address this large accumulation that occurs and that ultimately leads to cell death. We wanted to know whether this in vitro observation was actually true in other model systems. And so we actually looked in cells, in mouse tissue, in human organoids, as well as in a slice of the kidney biopsy from a patient with the disease. And in all cases, we observed specific colocalization of the mutant protein with this TMED9 COP1 marker. I'm not showing data where we actually had negative results with several other markers. Armed with this understanding, we wanted to see how, in the presence of the compound, we might be able to affect this localization. And indeed, what we found is that in the presence of the drug, in a time-dependent manner, we were able to efficiently push mutant protein through the secretory pathway and place it into the lysosome for degradation, which is shown here in the histograms, suggesting that the compound was essentially rerouting the mutant protein into the late secretory compartments. We wanted to understand this further in more mechanistic detail to try to understand how the drug might be doing this. And one of the first observations was that TMED9 was not only passively colocalized with the mutant protein, but in fact was also upregulated to handle the accumulation of the mutant protein in these tubular epithelial cells. This is shown here in the human organoid system. It was true in all of our other systems as well. And we were very interested to see that the drug was able to not only modulate, indeed clear the mutant protein from these tubular epithelial cells, but it was also down-regulating the TMED9 marker of the COP1 compartment, suggesting that there was a more active relationship between the compound TMED9 and the mutant protein. We wondered what would happen if we genetically deleted TMED9, and what we found was essentially that CRISPR-Cas9 deletion, effective deletion of TMED9, resulted in a phenocopy of the effect of the compound, suggesting that removal of TMED9 was sufficient to remove the brakes on the accumulation of the mutant protein and allow it to traverse the secretory pathway and be degraded in the lysosome. We wanted to know whether the drug is actually targeting TMED9 or a molecular complex that involves TMED9 in order to accomplish this. And so we performed a cell-based CETSA assay, which is shown here, which are some of the initial clues in this story that TMED9 is engaged by this compound, and this allows for the removal of its entrapment of the mutant protein, allowing it to be degraded in the lysosome. We are now actively involved in a lot of detailed mechanistic work, structure-based work, SPR type work to really nail down the exact binding of the compound to the TMED9 complex. And this is ongoing active work in the lab. All of this was interesting, but we wanted to know whether this was really true only in the setting of this particular toxic protein or whether there might be opportunities in other areas, in other disease areas. And of course, toxic proteinopathies are affecting several different tissues. For example, there's another autosomal dominant kidney disease caused by mutations in a protein called uromodulin that has a very similar presentation to MUC1. But there's also other areas such as retinitis pigmentosa, for example, mediated by rhodopsin mutations, or even Huntington's disease and many, many others. And so we wanted to understand the possibility that our compound might be having an effect. We did this by looking at an in vitro model for retinitis pigmentosa, and we were able to see that the compound reduces the mutant protein and also rescues cells from death. This was not true in Huntington's disease, which was actually very reassuring because mutant huntingtin actually does not accumulate in the secretory pathway, but instead accumulates in the cytoplasm and or the nucleus. And so this acted as an appropriate negative control for the fact that our compound is most likely targeting the secretory pathway. Since we're running short on time, I will just quickly run through this. I think we basically discovered a new tool that allows us to probe the secretory pathway and its ability to modulate the accumulation of misfolded proteins. And we think that this is a tool that can allow us to understand several toxic proteinopathies and really understand how cells are able to deal with the accumulation of these mutant proteins over time. Of course, we hope that this can ultimately inspire not only the deeper cell biological understanding of these diseases, but ultimately result in therapies, targeted therapies that can help us address these diseases. And with this, I want to thank everybody who works really hard in the lab to make this happen. And in addition, our patients who very generously donate their samples to make this research possible, as well as all of our funding agencies and numerous collaborators throughout the world. Thank you very much for your attention.
Thank you, Anna. Very interesting. And now, last but not least, Paul Workman. Paul, thank you for joining us today.
Hi, Mishtu. Can you hear me okay?
Yeah, perfect.
Great. Mishtu, thank you very much for the invitation. Thank you to all the organizers who put this together. And thanks to the attendees for joining. So you've heard great presentations then from Dan and Anna about different aspects of chemical probes. My main interest is in the discovery of new drugs for cancer treatment, and I do this at the Institute of Cancer Research in London. And aspects of the work that we do in drug discovery is actually done in the CIUK Cancer Therapeutics Unit, which I used to direct for a long time, and my lab is still part of that. So I'm really interested in using chemical probes to figure out and understand molecules that might be potential targets in cancer, and then to use them to validate targets, also to use them as pathway compounds to determine whether there might be a therapeutic opportunity with a small molecule related to the to the chemical tool. And in addition to my own research, I'm really interested in helping to the community, if you like, to improve the use of chemical probes by biologists. I don't know how many of the audience are more chemistry-focused chemical biologists or biological chemical biologists, or even straight people interested in doing biology with chemical tools. But I'm really interested in trying to encourage best practice, trying to encourage the selection of good tools by biologists in their experiments. And I do that by writing some reviews that I'll show you about, by giving talks like this, but also by building resources with my colleagues who I'll tell you about that will help what I think is a major problem in our community, which is the use of inappropriate chemical compounds for biological experiments. So that's my introductory comments. Here's my disclosures. I think the main things to highlight is I'll be talking about, if you just look at the 4th bullet point, I'll be talking about our PI3 kinase inhibitors, our CDK8/19 inhibitors, and our CHEK1 inhibitor. And you can see the disclosures that correspond to those particular projects and compounds. So just to give you an outline of what I'm going to cover, I'll talk about, again, maybe for students or postdocs or those who are new to this field, really to try and illustrate why it's important that high quality chemical probes, I stress high quality, are so important in biological research. And I'll take you through what we mean by high quality. What are the properties of chemical probe molecules that make them good or bad, and the current challenges that we face with their misuse, very common misuse and contamination of the literature because of that. I'll take you through some use cases from our own lab, and an example I particularly want to highlight in view of recent publications from elsewhere is the use of validation of chemical probes by using CRISPR knockout cell lines for the putative target of those compounds, and to show that you can really use that in the same way that Shelser and colleagues have pioneered recently. And then finally, as part of this outreach and trying to improve the community use of chemical probes, I'll talk about the resources that I and my colleagues have been developing to do that. So just by way of background, and again, this is targeted mainly to maybe newcomers to the field, and apologies to the experts who I'm sure are here as well. So the reason why chemical probes are so important is that they're valuable reagents to investigate biological processes and disease mechanisms and to validate drug targets. And of course, they provide complementary orthogonal methodologies to genetic perturbation through controlled modulation of target activity by modulating function rather than removal, and allowing dose-response experiments and that type of modulated perturbation, but very much as an orthogonal methodology alongside the genetics. And as I mentioned before, there's really incredibly widespread use and abuse in the literature of poor quality chemical compounds. And these range from the frankly promiscuous, insoluble or highly reactive, or just generally multiple frequent hitters in screens. And you'll see multiple papers in important journals that claim a chemical probe in what turns out to be a highly promiscuous molecule. So we've got to be careful about that. So we need particular care to be taken in terms of potency, selectivity, cell permeability, and importantly, evidence of target engagement and modulation in cells. And I think communicating the need for target and for careful and critical selection of chemical probes is really crucial. And I'll just show you an example from a paper that we published in Nature Chemical Biology with our colleagues in Merck in Darmstadt, from where we discovered by chemical proteomic screening and then chemical proteomic technologies these tri-substituted pyrimidine inhibitors of CDK8/19. And you can see in this little chart on the right-hand side that we're going from showing binding, in this case by physical methods, by doing a crystal structure to show that absolutely the target is binding by a defined mechanism in the target molecule. Then by using cell-based biomarker assays, in this case, we're using STAT1 phosphorylation as a readout of CDK90 inactivity, and then finally we go into phenotypic assay, which is the xenograft response in a tumor in an animal. And in that way, we can follow in an audit trail that everything is tying up together. And by using active and inactive compounds as controls and so on, that's also really, really valuable. So a slide that I often show in terms of choosing your chemical probes wisely is this one, where this is, you remember your Indiana Jones movie, this is the Grail Knight, and he gave the advice that you need to choose wisely, and we know that if you don't do that, there were devastating consequences, and if you did choose wisely, as Indiana Jones did, then you do a lot better. So I think one of the issues, the movie's not running, but not to worry, this is Whoopi Goldberg doing the Hallelujah. And I use this to say that I think one of the things that we've seen in the field is that we've been preaching to the choir. I think all the chemical biologists on this call will know many of these messages that we're trying to get over to the biological community. But we're failing, and we need to find better ways to make sure that the whole community, not just those who are chemically experienced, can make the right choices about chemical probes. And I care deeply about that. And we wrote with Julian Blagg, we wrote a review in focusing on the cancer angle in Cancer Cell, and you can read that if you want to hear more of our detailed thoughts on this. But it's all about best practice. What is the best practice for using chemical tools? It's about the good compounds, potent, selective, permeable, working through design mechanisms, and then the bad and frankly ugly molecules that we see, as I say, frequently being published. And here's some examples from great review article by Jonathan Beale and colleague Walters in showing the sorts of quite horrible chemically reactive or otherwise nasty molecules that you will find coming out of screens and being used in assays and in big papers. And it's not just, of course, that some compounds are broadly active, but they may just have somewhat differential activity on, say, kinases, inhibiting 2 or 3 other kinases from the target. But if they're crucial kinases, then that spoils your experiment. If those other kinases are also having a function in your system. And we just saw an example actually published in Nature of, and they did a great crystal structure of with other teams have done this, but the COVID-19 cysteine protease, the so-called MoPro, and this Nature paper that's just come out, where they said our results demonstrate the efficacy of the screening technology for the rapid discovery of drug leads with clinical potential. Now, the compounds that came from these screens that they said are leads with clinical potential are shown here. And all of the chemists and chemical biologists in the audience will know that these are all what are called Payne's compounds. So the selenium compound, epsilon, is a hitter in multiple assays, very broadly applicable. These two compounds in the middle, they're covalent reactors. You can see that the bottom one on the left, chiconin, is a deeply red-colored naphthoquinone, which is going to be redox active, and the other compound on the bottom right is a thyrodoxin reactive redox active compound. So this is the type of observation that these don't represent chemical leads. They don't even represent proof of concept because they're broadly acting indiscriminate compounds. And there are many, many examples that we could show on this. So here's an example of a study from a great Wellcome Fellow with his own funding, Albert Antolin, who is co-mentored by my computational colleague Bisan Lazikani. We set out to look at the possible off-target effects of PARP inhibitors on kinases. So we looked at these 4 PARP inhibitors that have been FDA approved. They have differences in PARP family inhibition profiles. I think I can show you that. You can see the different profiles that they have in those kinome trees there. They also have different activity as shown here with their chemical structures on the multiple members of the PARP family and the tencarases. So we did a comprehensive computational and then kinome screening assay using a binding assay, and we found that widespread binding to kinases. We found using a catalytic radiometric assay that niraparib inhibits DYRK1A and rucaparib inhibits DYRK1B, also CDK16 and PIM3 kinases, all at clinically achievable submicromolar concentrations. And then we moved on to test in intact cells. That's gone back. So there we are. Here you can see the activity of niraparib and rucaparib confirmed in binding assays in live cells using the nanobret technology with EC50s around 200 nanomolar. So these are the first reports of submicromolar kinase inhibitors in live cells. These are at clinically achievable concentrations. Now, we published this paper as a sort of prototype of the types of approaches that could be taken to look at off-target effects. We haven't yet followed up the pharmacological significance, but interestingly, you can see that olaparib, one of the clinically approved compounds, has no kinase activity for these target kinases shown here, and indeed was inactive in all the other kinase assays. So big differences in the off-target effects. And all of these compounds, several of them are used as chemical probes, and yet they have these different kinase activities. So just an example of, these are not promiscuous, these are not horrible compounds, these are just compounds where you have to be aware of what the off-targets might be. Now just very quickly, because we published this work before, again, this is for those who are new to the field, and I often get asked the questions by biologists, well, I understand what you're saying. Clearly, I want my chemical probe compounds to be high quality, but I don't know how to do that. And of course, the answer is not to go to Google, not to go to the vendor catalogs, but rather to go to trusted sources and look for features that are summarized here. So chemical features like stability, solubility, permeability. In terms of potency, you want high biochemical potency, cellular, less than 100 nanomolar, probably less than 30 nanomolar. Typically, you want cellular activity to be not higher than ideally 5, possibly 10 micromolar, ideally less than a micromolar, otherwise you run risks of non-specific effects. You want to do structure-activity relationships to understand how the compound's working. You might want the compound to be active in vivo. Then you've got, in the next batch of parameters here, you've got selectivity criteria. You want 50 to 100-fold ideally against the against the targets that you don't want to hit. You want to be as clean as possible. You also need to consider the biological context. And here are some just do's and don'ts from the Cancer Cell article. I'm not going to go through these, but people have contacted us and said they do find, biologists who are working with chemical probes do find these do's and don'ts advice quite helpful. So I'm going to run through some quick examples from our lab. This is all work done by my close colleague, Paul Clark, with whom I've worked for many, many years in our lab. And this is, in fact, a particular project where he led the study, and it was to look at the problem of drug resistance, we were seeing that MEK PI3 kinase combination of drugs was very active in KRAS colorectal cancer cell lines. But even with the combination treatment, we were seeing resistance generally being developed through the RAS, RAF, MEK, ERK pathway. We could, and we published this in an oncogene paper recently. And here, by the way, you can see the structure of pictulisib, which is a chemical probe for PI3 kinase, and also a drug that this is a compound we discovered at the ICR in collaboration with Pyramid and licensed to Genentech and went into the clinic. And we're using it here as a chemical probe. We had an inactive analog as well. It was very helpful where the effects were not seen. And here we also used another chemical tool. This is navitoclax, which is a BCL2 inhibitor. And we found that combining this compound after screening for BCL2 family members, we found that this drug, when combined with the other two drugs, was able to block drug resistance. So at the same time, we were using drug treatments, but it's equally well using these as chemical probes to explore mechanism. So this is an interesting, potentially relevant clinical combination. This is an assay this is a screen, by the way, that was done by a fantastic postdoc, Qi Zhang, in the lab in collaboration with the Mitnack, Sibylla Mitnack group at UCL. And here we were looking at tackling the problem of resistance to CDK4/6 inhibitors. CDK4/6 inhibitors are amongst the most exciting new drugs for ER-positive breast cancer patients in a decade, probably the most exciting. But drug resistance develops relatively quickly, and also some patients don't respond, and patients with other tumor types that you'd expect would be responsive don't respond. So we ran a screen looking for an siRNA screen initially with this. You can see at the top the assay that we used. It was a image-based analysis looking for mechanisms that would lead to this resistance mechanism that we see, which is due to activation of CDK2. And then we wanted to find those genes which, when knocked down, would reverse this resistance. And you can see in the shading and the stars there that we came out with highly scoring genes there were MET and FAC. And then we used the chemical compounds to see if we could phenocopy the genetic synergy, if you like, between MET or FAC knockdown in these cells, overcoming resistance to the CDK4/6 inhibitor. You can see that we use two different, here's palbociclib, by the way, the structure of that. We use two different CDK4/6 inhibitors at the top and two different MET inhibitors. The FAC data is not on this slide. And here you can see on the right-hand side that you get a synergistic interaction. So again, we've been using, an important message here is the use of two different chemical classes of inhibitor for both the CDK4/6 side and also the MET side, and then in addition, two different FAC inhibitors. Using two different inhibitors and finding the same result gives you greater confidence in the result. So we hope that this is a combination that might be tried clinically to slow down resistance in breast cancer patients with being treated with palbociclib. But it's also a nice experiment in chemical biology as well. Here's something that just came out in cancer research from the work of a fantastic graduate student, Rebecca Rogers, working between my lab and previously was in Michelle Garrett's lab, and then we did the work in combination. And we were asking the question, so CHEK1 inhibitors are really have exciting potential to exploit the absence of cell cycle checkpoints in tumor cells. And by inhibiting CHEK1, you have potential activity through exploiting replicative stress in MYC-driven cancers, for example, but also to potentiate the effects of a cytotoxic inhibitor like gemcitabine. And here again, we ran a screen as shown in the previous type of slide, previous type of experiment, but it was a viability screen. And we were looking for genes which, when knocked down using a druggable genome siRNA screen, would potentiate the effects of a CHEK1 inhibitor. In fact, the CHEK1 inhibitor here is the one we discovered at the ICR and then licensed to Sierra Oncology and is currently in phase 1 trial. It's a highly selective drug and a highly potent chemical probe for CHEK1. Its structure is shown here. This is a second CHEK1 inhibitor that we use just for a different chemotype. And you can see the hits here are all in the family of subunits of the B family of DNA polymerases. And so in order to get chemical validation of the observation that this was functionally, to get evidence of functional activity here, we then repeat the experiment using either a phytocolin, which is a well-known natural product that's an inhibitor of the B family of DNA polymerases, and also this retinoid compound. So here's a phytocolin, and here's this retinoid compound that we discovered in the literature also to have that activity. And you can see we could fully recapitulate the shift in the dose-response curve to the left, shows that by both genetic knockdown and by chemical manipulation, perturbation, with 2 different classes of inhibitors in both types, we robustly phenocopy this effect in multiple cell lines. And so we see this as sometime in the future, maybe when there's a B family polymerase inhibitor available, this could be an interesting clinical combination, another nice demonstration of validating a genetic discovery with small molecule chemical tools. This is, I think, the last example I'm showing, which is using CRISPR knockout. And again, this is work led by Paul Clark in the lab, using these 3,4,5 tri-substituted pyridines that I mentioned at the beginning of the talk in that audit trail. We were looking to see whether we would, the rationale summarized in the bullet points you can read quickly through there. By chemical proteomics, we identified the CDK8/19 inhibitors of Wnt signaling, actually, and then we wanted to ask the question: does properties like the molecular properties, gene expression profiling, and other features, does it reside in CDK8? Does it reside in CDK19? Or might it even be an off-target effect? We already knew that our inhibitors in this series from an eLife paper were not only highly potent, but hugely selective in kinome profiling and transcriptional profiling. What we found, here's an example of soft agar growth response. We saw that the most effects of CDK8/19 inhibitors are lost when you knock out CDK8, but not when you knock out CDK19. So here's a soft agar growth assay in the middle of the slide. You can see that the parental line responds like the black line. The blue line, if you knock out CDK19, they still respond, suggesting CDK19 is not the target. If you do a CRISPR knockout of CDK8 and test the drug in that line, you've lost activity. And so that strongly suggests that CDK8 is the primary target, not CDK19. Obviously, the double knockouts behave like the CDK8 knockout. All the other features of the drug, of the CDK19 inhibitors, behaved in the same way with the knockouts. Interestingly, apart from when we did an in vivo xenograft assay. And here you can see that there's full activity with the individual CDK8 and CDK19 individual knockouts. It's only when both CDK8 and 19 were knocked out together that you lose the anti-tumor activity in vivo. All the in vitro assays showed that the primary effects were due to CDK8 inhibition. But in this single xenograft model, we need to look in other models, suggests that for some reason in vivo, CDK19 also plays a role. This is also, I think, a nice use of this technology developed by Chelsea and used by others, that you can use knockout lines to ask whether your drug is really working through the intended target. So I want to move on in the final section now to this more educating side of what I talked about, the need to make it easier for biologists to find tools that are the best ones to use, not the oldest and most cited, not the one that the vendor catalogs necessarily are selling, and not prioritizing for quality. But what are the best ones that you can use? And if you're interested in this at all, you might be interested to read a recent review that I published with Albert Antolin and Bisan Lazikani, who I mentioned before, my fantastic colleagues working in this area. And I can just get that to, yeah. So we feel that the way to do this is to use resources that are either expert peer review resources, and I was one of the people involved in the early founding and became a director of the Chemical Probes portal. This is a great resource if you want to find out for your target what the best chemical probe is. Then you can simply go to this resource, chemicalprobes.org. You can, for those targets that have probes reviewed, you can find the expert peer review, and you can find compounds that are recommended and those that are not recommended. And another way to do this is to use Probeminer. This was developed by Albert and Bisan with myself, with involvement from Ian Collins, who I mentioned before. This is a complementary large-scale computational analysis resource built on Kansar and Kemble, other resources that Bisan was the architect of. And this is entirely complementary to the Chemical Probes portal, because you're not asking for an expert review in this case, you're asking the computer to rank using statistical methodology for the best compound, using either preset features or total resources that you can set. So these are tools showing just on the right-hand side here. You can read the review, how suggesting these tools are used together. And we also publish here, I won't go into this in detail, but we publish a decision tree. So again, if you're a chemical biologist looking to see, for my target, what drug should I use? You can go down this decision tree and see if there are probes in the portal, see if there are probes in the Probeminer, see if both of those work together, and you can just work through this. And it kind of gives you if you're not familiar with how to think about chemical tools in this way, it gives you an aid to hopefully get to the right point. And I'm just showing here 3 examples. I realize I'm running out of time, so these examples are in the paper that I showed you. There's an example, for example, if you're looking for PI3 kinase inhibitors, if you're happy with a pan PI3 kinase inhibitor, you can use pictolizib, which is in the portal as shown here. If you really want one that's highly selective for PIK3CA, that's the alpha isoform, then it's better to use this other probe that's shown here. Then I'll show you an example. Just being a bit slow to click through here. So can you still hear me? I'm sorry, I've lost the, Mishtu, can you hear me?
Yes, I can hear you, Paul.
I've lost the images now, so I'm just going to conclude. If you don't mind just clicking me through for the next couple of slides, and then I'll close, because I've just lost the image. So I just want to go back again. This is the messages for the biologists and not for the experts, but we're going back to the Grail night again here about choosing wisely on the slide that's called Choose Your Chemical Probes Wisely. It's all about, if you're selecting your target, you need to be aware of the requirements of your assays in your biological system. If you want to get to the best probe, use a combination of the expert curated resources like the Chemical Probes portal. Use the statistical methodologies like Probeminer, and there are other resources listed in the paper. Avoid the historical bias. Avoid what maybe the vendor catalogs are suggesting, and look for what the experts and the computational resources direct you to. Of course, talk to medicinal chemists and chemical biologists. Next, do become familiar with the limitations of your probes. You can use probes that are not ideal. Oh, thank you, it's back. You can use probes that are not perfect. Sometimes you have to. But be aware if you're inhibiting another kinase, or if you're using a PARP inhibitor, maybe you're inhibiting another kinase. use knockout cells to see if your compound still retains activity when you've knocked out the target. If it does, then it's not working through the intended target. I showed you the CDK19 experiment where we showed that our drugs were working by the right target. Clearly, you need to be able to obtain the compound commercially, but there are now resources that are being developed where you can obtain really high quality chemical probes from free of charge from some of the consortia that are now getting together to drug the proteome and make those tools available. Always use more than one chemotype, always use an active compound if you can, and use that system alongside genetic knockouts. So just to conclude, these are the people, my great colleagues on the chemical probes, Portley and Collins, Julian, Bisan Alazikani, Albert Antolin, Suzanne Muller-Knapp, Aled Edwards, Cheryl Arrowsmith was very actively involved and still a great proponent of chemical probes as well, and many others. I went through the people who were on the projects that I described to you. So I'd like to thank all of them. I'd like to thank our funders, Cancer Research UK and Wellcome Trust, also Merck for the CDK19 project, all the collaborators and great students and postdocs. And finally, I want to thank you for attending and for listening. I hope that was helpful.
Thank you, Paul, for this nice presentation, and many thanks to all our speakers for sharing the exciting discoveries. We are now going to take, in the interest of time, 3 questions to go over some that our listeners have submitted. So the first question, this is for Anna. Does the MUC kidney disease have altered glycosylation of MUC1? What are the significant changes?
Yes, this is an interesting question that we are actively investigating. MUC1 is heavily O-glycosylated, and that happens around that VNTR region. And so I don't really have an answer. It's an excellent question, and one that we're actively looking at right now to look at the patterns of glycosylation, and in fact, to look at whether any changes in those patterns may actually affect the trafficking of the mutant protein versus the wild type. Thank you for the question.
Thanks, Anna. So this is a question for Paul. Do you think the selection during knockout generation would obfuscate whether drug activity is associated with the known target?
Yeah, that's a good question, and thank you for it. So it is something that you have to be really aware of. Clearly, if you're, as you rightly say in your question, if you're selecting a clone in which you've knocked out the target, then there is the possibility of, that cell line has of course, become no longer dependent on that target, but could have other changes as well. So using multiple clones, I mean, the key thing I think is that if you take out a target, let's say it's CDK8, you think your target is CDK8, you knock out CDK8, so that cell line is presumably no longer dependent on CDK8. Then you put on your compound and you still have activity. Clearly your compound is not working through CDK8. And if you have lost activity, then you have a good idea that you're selective. So I think you have to be careful of what you've just mentioned, but I think using these assays to ask the right questions can still be very insightful. Thanks for the question.
Thank you, Paul. So the next question is, which would be our last question, what would be the most appropriate chemical probe for interrogation of phosphatases as a complementary chem bio technique compared to kinases? Either Paul or Anna, you could answer.
Sorry, could you repeat that again? I'm not sure I got the question.
What would be the most appropriate chemical probe for interrogation of phosphatases as a complementary chem bio technique compared to kinases?
So phosphatases are in the highly tough to drug. We certainly worked on these in the past, and they are just incredibly difficult targets to produce small molecule inhibitors for. I think I'm not an expert myself in phosphatases. I don't work on them now, but I've heard that a good approach to take is to look at those phosphatases that require protein-protein interactions to deliver their biological function, and look for molecules that might block the interaction between the phosphatase and a requisite cofactor protein. I think that's, off the top of my head, I'm not familiar with compounds that really would be good chemical tools for phosphatases, but I may be a little bit behind in that area.
All right, thank you, Paul. With that, I think we have come to the end of our session, and we are very grateful to our sponsor, Cayman Chemical, for the contributions that made this webinar possible. We would also like to thank you, the audience, for tuning in today to learn more about chemical probes as essential tools for biological discovery. If you missed anything during this webinar or would like to listen to it again, a recording will be available shortly on the cell.com website. If you have any comments about this webinar or suggestions for future topics, we would love to hear from you by email at cpwebinars@cell.com. Keep your eyes peeled for our upcoming CellPress webinars and related Cell Symposia at www.cell.com/cellsymposia. Also, don't forget that I'm the editor of Cell Chemical Biology. For any journal-related matters, I would love to hear from you by email at chembio@cell.com. Thank you very much.